My responsibility was to discuss three separate abstracts. One dealt with signaling cascade downstream from the EGFR pathway. One was a study of looking at rare EGFR mutations, in particular exon 20, which we don't see quite often, and we're not quite sure how to treat those patients. And the last one was a study from Japan looking at potentially coming up with genetic factors that could explain why more Asians get an EGFR mutated non-small cell lung cancer. So the first study was the more complicated one. Several different signaling pathways were interrogated by Dr. Rosell, and he showed very elegantly that by bringing in 2 or 3 different tyrosine kinase inhibitors, you can block certain key pathways that, at least in the cell culture and animal model, gave pretty interesting results in terms of tumor suppression. And the main topic was, well, can we translate this into a clinic? And are there other examples of similar experiments, which is what I was able to sort of discuss. The second one was by Dr. Reese from UC Davis, where he looked at genetic profiling of multiple tumors, digging out those exon 20 mutations. He showed that there are multiple different exon 20 mutations, in fact. And then he used a PDX model, which is taking a tumor from a patient, putting it in a mouse. We think that mimics exactly what happens inside of a patient, because the tumor directly comes from a patient. And then again, through a series of animal experiments, he was able to show that a combination of 2 separate EGFR pathway inhibitors, an antibody and a TKI, is actually pretty effective in suppressing tumor growth in these animals. And his idea now is to take that combination into a clinical trial, which, as I understand, is already ongoing. So we talked a little bit about some of the difficulties with combining these drugs. We have a prior experience from another TKI and antibody being mixed in a clinical setting, combined together and giving a little bit of a side effect. So the question was, is this really a plausible way of proceeding for these rare EGFR mutations? And the last one was a little bit of outside of my immediate area of expertise, but Japanese group who's done a lot of work with genome-wide association studies and looking for different HLA haplotypes, trying to explain again why do we see so much more EGFR mutated lung cancer in the Asian patient population, which now from multiple studies we know that is the truth. They have a lot more of the EGFR mutation. So they identified a particular HLA that seems to be associated at a higher level with EGFR mutation, and the gist of my discussion was, what do we do with the information? Now this adds to our prior knowledge, but it raised a very interesting theoretical point in that this particular allele is in an HLA class 1, where we know there's a lot of antigen and T cell recognition pathways. And is it really possible that somehow the immune system is involved in this whole process? Now, how to link it to induction and tumorigenesis is something that we don't know yet, but I thought it was an interesting study from that point of view.